it's time to hear Emma. So we change. We go to PRV3 calcium channel. Okay.
Okay, so good afternoon, everyone. I'm really happy to be here today to present my thesis work, which focuses on the role of the TRPV3 calcium channel in psoriasis. So as you know, psoriasis is a chronic inflammatory skin disorder with a high worldwide prevalence, located between 0.09 and 11.3%, which is characterized by the appearance of localized and delimited erythematosquamous plaques on the body. At the histological level, psoriasis is characterized by epidermal hyperplasia, which is due to keratinocytes hyperproliferation, but also to some differentiation defects of keratinocytes. But what leads to the appearance of those psoriasis-related features? What really happens in the skin of patients? So the triggering events, which consist in interactions between environmental factors and genetic susceptibilities, leads to the persistent activation of Th17 lymphocytes. Those Th17 lymphocytes can produce IL-22 and IL-17A that directly act on keratinocytes. Keratinocytes will start to hyperproliferate, and in response, they also produce antimicrobial peptides, chemokine, as well as several pro-inflammatory cytokines, including IL-17A. So here you can see that keratinocytes also play a key role in the inflammatory response of psoriasis. And among the protein that you will find expressed by keratinocytes, there is TRPV3, which is a calcium channel abundantly expressed by keratinocytes and by keratinocytes only. The TRPV3 protein is formed of 6 transmembrane domains, which self-assembles into homotetramer with a pore in the middle, allowing the calcium ions to enter the cells. There are several ways to activate TRPV3 channel. The first one is heat, with the thresholds of activation between 32 and 39 degrees. But you can also activate TRPV3 with some natural compounds or synthetic compounds like carvacrol or 2-APB. TRPV3 can also be inhibited by using some specific antagonists like IAB, for example, which is the one I use in this study. So there are some evidence that TRPV3 can be linked with cutaneous inflammation, as here, for example, where a gain-of-function mutation in the gene encoding TRPV3 can be the direct cause of the appearance of a chronic inflammatory skin disorder called the Olmsted syndrome. In this disease, which shares similarities with psoriasis, TRPV3 is overexpressed in the epidermis of patients compared to healthy subjects. Moreover, as it is a gain-of-function mutation, here TRPV3 channel is also hyperactive. It is also the case in atopic dermatitis, so the first chronic inflammatory skin disorder in the world, where TRPV3 expression is increased in the skin of patients compared to healthy subjects, and where some calcium imaging experiments showed that the TRPV3-dependent calcium influx was increased in keratinocytes derived from AD patients compared to healthy controls. So we just saw that TRPV3 was overexpressed and hyperactive in the skin of patients suffering from 2 chronic inflammatory skin disorders, the Olmsted syndrome and atopic dermatitis. However, to date, there is no evidence that TRPV3 could be implicated in psoriasis, which is also a chronic inflammatory skin disorder. This is the whole point of my thesis, to know if psoriasis is TRPV3, is involved in psoriatic inflammation. To do so, we divided our work into 3 main axes. The first one focuses on TRPV3 expression in psoriasis. The second axis aims to see if the TRPV3-dependent calcium influx is modified in psoriasis conditions. And the third axis aims to decipher some TRPV3-dependent inflammatory mechanisms in the context of psoriasis. To do so, we have different models. The first one are human skin biopsies from healthy donors or from psoriasis patients. The second model is the imiquimod-induced psoriasis-like mouse model, which consists in topical administration of imiquimod, an active substance able to induce a psoriasiform dermatitis in mice. So we apply imiquimod on the back skin and on the ear skin of mice for 4 consecutive days, and our control mice are treated the exact same way, but with Vaseline. Finally, we also have a psoriasis-like cellular model consisting in the culture of human primary keratinocytes derived from healthy donors that we put under a psoriasis-like inflammation by using a cocktail of 5 pro-inflammatory cytokines known to act in synergy to mimic a psoriasis-like phenotype in vitro. And this is called the M5 cocktail. So first, for TRPV3 expression, we first looked at this in the human psoriatic epidermis by performing TRPV3 mRNA in situ hybridization. And you can see on those microscopic images that the red signals corresponding to TRPV3 mRNAs seems more intense in the epidermis of patients suffering from psoriasis compared to healthy controls. When we quantified this, we saw that TRPV3 was 3 times more expressed in the lesional epidermis of patients with psoriasis compared to non-lesional epidermis and to the epidermis of healthy controls. At this point, we didn't know the exact location of TRPV3 overexpression in the epidermis of patients with psoriasis. When we looked at the images, we can see that the signal for TRPV3 mRNAs is more located in the upper layer of the epidermis, so more in highly differentiated keratinocytes. But at this point, we weren't very sure. So I also looked at TRPV3 expression, but this time in cultured human primary keratinocytes during the whole differentiation. To do so, I cultivated the human primary keratinocytes with or without the M5 cocktail to induce the psoriasis-like inflammation in vitro. And I performed RTQ PCRs at different times during differentiation. You can see here that we found that TRPV3 was overexpressed in the keratinocytes that were cultivated under psoriasis-like conditions compared to untreated keratinocytes, and this at day 7 and day 9 of differentiation, which corresponds to the moment when the expression of involucrin, so a marker of terminal differentiation, is at the highest amount. So those results here confirms what we saw in the skin of human patients with psoriasis, so that TRPV3 is overexpressed and that its overexpression occurs more in highly differentiated keratinocytes. Then I looked at the expression of TRPV3, but in the skin of the imiquimod-induced psoriasis-like mouse model. And here you can see that TRPV3 was found to be overexpressed in the skin of mice that were treated with imiquimod compared to Vaseline controls. And this was the case only in the ear skin and not on the back skin. But here again, it confirms TRPV3 overexpression under psoriasis conditions. After looking at TRPV3 expression, we were also interested in TRPV3-dependent calcium influx in psoriasis conditions. And to do so, to look at this, we performed FURA2-AM calcium imaging experiments, still on cultured human primary keratinocytes that were treated or not with the M5 cocktail. So just to explain, FURA2-AM is a calcium-sensitive indicator which can enter the cells. And it displays an excitation peak from 390 nanometers when the probe is free and not bounded to calcium in the cells, to 340 nanometers when calcium enters and bounds to the probe. As a result, both free probe and calcium-bounded probe emit fluorescence, which allows the ratiometric measurement of the relative fluorescence between both wavelengths. As a result, when the fluorescence ratio of 340 over 380 increases, it means that you have an increase in the intracellular calcium. So with that, we stimulated keratinocytes with TRPV3 agonists, and following TRPV3 stimulation, you can see that the TRPV3-dependent calcium influx seems more intense in keratinocytes that were cultivated under psoriasis-like conditions compared to untreated keratinocytes. To be sure that those responses were really TRPV3-dependent, we also added conditions where we treated the keratinocytes with a TRPV3 antagonist, and when this antagonist is present in culture, even if you stimulate TRPV3 with its agonists, you can see that there is almost no calcium that enters the cells, which means that those responses here were really TRPV3 dependent. Then we calculated the area under the curve, which corresponds to the total amount of calcium entering the cells following TRPV3 stimulation and during the entire response. And you can see that the TRPV3-dependent calcium influx is significantly higher in keratinocytes that were cultivated in psoriasis-like conditions compared to untreated keratinocytes, thus suggesting an hyperactive TRPV3 channel in vitro in this model under psoriasis conditions. Finally, the third axis aimed to decipher some TRPV3-dependent inflammatory mechanisms. And to do so, we came back to our imiquimod-induced psoriasiform mouse model, but this time we performed imiquimod treatments in both wild-type and TRPV3 KO mice to see if TRPV3 KO mice would develop psoriasiform dermatitis in the same way as wild-type mice, and if not, what would be the parameters that changed. And here, the first thing we noticed was that following imiquimod treatments, TRPV3 KO mice seems to present a less severe psoriasiform dermatitis compared to wild-type mice. And to quantify this, we calculated a PASI-like score, which is a clinical score used to quantify psoriasis severity based on the visual evaluation of skin redness, thickness, and desquamation. And you can see that both wild-type and TRPV3 KO mice present an increased PASI score following imiquimod treatments compared to vaccine treatments, suggesting that both genotypes develop a psoriasiform dermatitis following imiquimod applications. However, the PASI score for the TRPV3 KO mice was significantly reduced compared to wild-type mice, suggesting that TRPV3 could positively regulate the severity of the imiquimod-induced psoriasis in this model. And to go a little bit further, I performed some skin sections, and you can see that in wild-type mice, after imiquimod treatment, there is an epidermal hyperplasia, which is normal and which was expected. However, the thickness of TRPV3, of the epidermis of TRPV3 KO mice following imiquimod treatment seems really reduced compared to wild-type mice. So we did several measurements on other individuals, and you can see here that epidermal thickness of TRPV3 KO mice after imiquimod treatment was significantly reduced compared to wild-type mice. So here, this suggests that TRPV3 could regulate epidermal hyperplasia following imiquimod treatment. Finally, we looked at the expression and secretion of IL-17A, so this major psoriasis-related cytokines. And you can see that TRPV3 KO mice expressed 50 times less IL-17A compared to wild-type mice following imiquimod treatments, and that IL-17A secretion also tends to be decreased in TRPV3 KO mice compared to wild-type mice after an imiquimod treatment. So here, when TRPV3 is absent, there is less expression and secretion of IL-17A, which could potentially explain why TRPV3 KO mice develop a less severe psoriasiform dermatitis in this model. Finally, to come closer to the human and to try to corroborate the results we obtained in mice, we tried to generate 3D model of psoriasis-like reconstructed epidermis in vitro, still by using human primary keratinocytes and by generating reconstructed epidermis that were treated or not with the M5 cocktail. You can see here that reconstructed epidermis that were cultivated under psoriasis-like conditions have an increased thickness compared to control epidermis. And more interestingly, when TRPV3 is inhibited under psoriasis-like conditions, the thickness of the reconstructed epidermis reduces to come closer to the thickness of control untreated epidermis. Here, this result confirms what we saw just before in mice, suggesting that TRPV3 regulates epidermal hyperplasia in a psoriasis context. I also looked at the secretion of IL-17A and IL-8 pro-inflammatory cytokines involved in psoriasis in this model. And if the M5 cocktail induces the secretion of those pro-inflammatory cytokines, when TRPV3 is inhibited, there is a significant reduction in the secretion of those cytokines, meaning that TRPV3 could also regulate the secretion of pro-inflammatory cytokines, at least in this model. So to summarize, we saw that TRPV3 expression was increased in the human lesional psoriatic epidermis, and this overexpression was also found in our different models, so in human primary keratinocytes that were cultivated under psoriasis-like environments, and also in the skin of the imiquimod-induced psoriasis-like mouse model. We also showed that the TRPV3-dependent calcium influx was greater in human primary keratinocytes that were cultivated under psoriasis-like conditions, and that TRPV3 positively regulates psoriasis severity in the imiquimod-induced psoriasis-like mouse model, mainly by regulating epidermal hyperplasia and the expression and secretion of pro-inflammatory cytokines. Altogether, those results suggest that TRPV3 could represent an interesting therapeutic target in psoriasis. However, there are still some TRPV3-dependent mechanisms that remain to be explored, notably the TRPV3-dependent signaling pathway that could be participate in psoriasis inflammation. To date, the only pathway known to derive from TRPV3 activation is the EGFR pathway. And this EGFR pathway is also known to participate in psoriasis pathogenesis, as its activation leads to the activation of some transcription factors involved in the regulation of genes, involved in cell proliferation and differentiation, and also in inflammation. However, the direct link between TRPV3, EGFR, and psoriasis inflammation, or in the defects of differentiation or proliferation observed in psoriasis, has never been made, which makes it a great avenue for further exploration of TRPV3-dependent mechanisms in psoriasis. So thank you all for your attention, and I will be happy to answer your questions.
Thank you. Is there some question for Emma? Oui. Corinne?
Thank you very much for this very nice presentation. I have a question regarding the expression of TRPV3. Do you know what is regulating its expression in non-genetic disease?
Not really. We don't really know. There is one protein that is known to regulate its expression and its activity. It's TMEM79. It's a receptor coupled to the G protein. And it's shown that this protein negatively regulates TRPV3 expression and secretion. So it would be interesting to go and see if the expression or this activity of this protein is decreased in psoriasis, and could explain why we have this overexpression of TRPV3 in psoriasis. And I also know that there are some microRNA that could present some sites that could target TRPV3. also. But
and if I may, I have another question about calcium.
yes.
Calcium usually it is buffered by different compartments. It is localized to different compartments. So you looked at the global cellular calcium concentration. Have you looked at the mitochondria or reticulum endoplasmic localization of your calcium?
No. Here, the experiments were performed with calcium in the extracellular in the buffer. So, and it's also important to note that here the experiments were performed in proliferating keratinocytes, and in proliferating keratinocytes, TRPV3 is located at the membrane. So here we just see the calcium that goes through the membrane, but I have no further information on if it comes more from the endoplasmic reticulum. Maybe we could perform the experiments also without extracellular calcium to see if the calcium could come also from the stores. But here I have no further information. It could come from the extracellular environment or from the stores.
Okay, but you can check you have some specific probes to look at if you have change in the calcium concentration, but I understand what you said.
Yes, okay.
Thank you.
Thank you.
Lionel?
Thank you for your talk. So can we go back to the results that you get in 3D model, please?
Yes, I don't have the hand on the computer
you don't
right
have the
now.
hand, so so my question is about TRPV3 and microflora. Exactly. So, because so many people speaking about psoriasis or any time or atopic disease, but so, and what about the microbiota and the regulation of them? So, do you test any kind of? So, first one, did you check the Staph aureus relationship with TRPV3? And no?
No.
And you plan to do that?
No, we don't plan to do that. No. No, yeah, I think it's...
So if I see Bérangère or Jérôme in a couple of hours, I can suggest to them to do that.
Okay.
Okay, thank you.
Thank you.
Marek? Yes. Your neighbor. Yes.
Okay, thank you for the nice presentation. I have a question about the knockout animals. So these TRPV3 knockouts, do they have any phenotype, and is this knockout, maybe I missed it, but is this a full body knockout? So do these animals lack the TRPV3 only in the keratinocytes or also on the sensory nerves?
Yeah, okay. So for your first question, the phenotype of TRPV3 mice, the only phenotype is when the mice are newborns. They have whiskers that are curvy, because TRPV3 is also implicated in hair growth and morphogenesis. But in adult mice, it's not the case anymore. And your second question, it's a knockout in the entire body. It's not tissue-specific in the epidermis. However, in mice, I don't think that there were demonstration of TRPV3 activity on the sensory nerves. It was described that it was expressed, but no demonstration of TRPV3, an active TRPV3 on sensory nerves. But it can also be expressed in some small vessels of the brains, and to remove this potential bias, we have the tissue-specific KO that is coming to the laboratory to have only the deletion in the epidermis.
Oh, okay, great. Thank you very much.
No other question? Thank you.
Thank you very much.
So now it's